Hello, I'm Ryan, an applications informatician with Oxford Nanopore Technologies. As a member of the applications group, I research, develop, and support software tools to enable experimental application scientists. In this video, I'll be discussing a novel extension of Oxford Nanopore's base calling algorithms. The applications group explores and demos uses that were previously difficult, if not impossible, with existing short-read, fixed runtime technologies. We exploit the natural advantages of nanopore sequencing on the MinION platform. It's portable, it's single molecule, it's long read, and it's real time. For more information about apps, stop by our posters or have a chat with one of the members of the group. During a sequencing run on the MinION, we measure the current flowing through each nanopore independently. As DNA strands pass through the nanopore, this current changes. The current level at each point in time corresponds to a specific set of bases in the pore, termed a k-mer. By matching the current level to its corresponding k-mer, we produce a base call. Nanopore's base calling algorithms are based on hidden Markov models, HMMs for short. HMMs rely on a statistical model of transition and emission probabilities. For example, consider a dishonest casino where the casino swaps in a loaded die. A transition is the probability that the casino changes the die. An emission is the probability distribution of the fair and loaded die respectively. Within the context of base calling, a transition is the probability of moving to the next k-mer, while an emission is the probability of a specific k-mer given the current level. Importantly, base calling relies on an unconstrained transition path. The maximum likelihood path is taken as the base sequence. In base calling, we allow all possible transition paths because we have no prior knowledge of the sequence. However, with prior knowledge, we can constrain the HMM. We call this approach mapping. Due to the constraint, mapping is orders of magnitude faster than base calling. As such, it is particularly useful for rapid strand identification. Custom HMMs are partially constrained HMMs that lie in the intermediate area between the wholly unconstrained base calling and the wholly path-constrained mapping. By constraining either the transition path or k-mer set, custom hidden Markov models allow the incorporation of prior biological information. Custom HMMs are assembled from modular blocks. Currently, these include calling, mapping, and junk blocks. These modular blocks may be assembled with different types of connections, including linear, repeatable, and branch structures. As we will demonstrate in a series of case studies, custom HMMs may be used to address a variety of biological questions. For example, consider a test for Huntington's disease. Normal and disease states differ in the number of repeats of a CAG trinucleotide repeat present in the Huntington's gene. For normal individuals, this is between 10 and 26 repeats. For clinically affected individuals, the number of repeats is generally greater than 40. So, how many repeats are there in the squiggle at left? We could base call the sequence, but we'd be discounting much prior knowledge. We know the flanking sequences, as the leader and hairpin adapters are standardized. We also know the trinucleotide repeat sequence, CAG, for the Huntington's gene. By applying our prior knowledge, we can construct a custom HMM to answer the biologically and clinically pertinent question, how many CAG repeats are there in the strand? For the template section, we construct a custom HMM for the Huntington's gene. First, we map to the known leader flanking sequence. Next, we map with a repeatable connection to an unknown number of repeats. Finally, we map to the known hairpin flanking sequence. An analogous custom HMM may also be constructed for the complementary strand. Digging deeper, we show a reduced 3-base k-mer representation. While both linear and repeatable mapping have fixed k-mer sets, in this case the Huntington's repeat, CAG, colored in red, they differ in that the repeatable mapping transition path is cyclic. Importantly, applying the custom HMM yields a single molecule estimate of the number of repeats contained within each individual strand. We applied the custom HMM to cell line genomic DNA samples derived from 3 patients, 2 with no phenotype and 1 clinically affected individual. Custom HMM yields reasonable estimates of the number of repeats achieved with single pass 1D data, about 100 times faster than base calling, in near real time. For more information on repeat sequencing with Nanopore technology, see the poster, Sequencing Short Tandem Repeats. For another example, consider cancer prediction. A recent study has linked the telomere length of circulating cancer cells to disease severity and progression. Containing kilobase stretches of simple repeats, telomeres are not addressable with short-read sequencing approaches. These limitations do not apply to nanopore sequencing. Considering data from a telomeric ladder with 100, 200, 400, 800, and 1600 repeats, we use our knowledge of the telomeric repeat to construct a custom HMM. At left, we consider only knowledge of the repeat. At right, we also include flanking sequences. The more biological information available, the more accurate our single molecule estimates. Not all repeats are homogeneous. Diseases such as the spinocerebellar ataxia family of neurodegenerative disorders possesses a complex repeat pattern spanning several kilobases. While these kilobase stretches of complex repeats are impossible to address with short-read sequencing, they are trivial with long-read nanopore sequencing. Again, we construct a custom HMM tailored to the biological question: what is the structure of the complex repeat in the sample? Unlike the Huntington sample, the flanking sequences are variable, so we base call instead of map. Additionally, if the order of the repeats were unknown, we could construct a different, branched custom HMM, shown at right. We identified the number of repeats for each subsection of the complex repeat. In part, owing to the previous difficulty of sequencing long complex repeats, the linkage between repeat and disease state are less well established for the spinocerebellar ataxia disorders than for simple trinucleotide repeat disorders. Custom HMMs are also useful for research purposes. For example, we wanted to know the length distribution of RNA transcripts in DNA-RNA hybrids. We constructed a custom HMM to map to the known adapter sequence and base call the RNA. By examining the length of the base called sequences, we determine the RNA transcript length. For more information, see the applications PechaKucha entitled Direct RNA Sequencing. Imagine the possibilities of applying custom HMMs. Applications such as monitoring somatic repeat expansions, real-time blood telomere length monitoring, and panel screening for neurodegenerative diseases. The possibilities are endless. The Applications Group thanks you for listening.